Every living thing on the planet stores genetic information in its DNA. Over the last 30 years, scientists applied several mutation technologies using DNA information in crop breeding to develop wide varieties in agriculture. Recently, genome editing technology, another milestone in bioscience, has been rapidly introduced worldwide as a breeding tool for crop development. Most seed companies use this technology or are highly interested in current trends. On October 23, 2012, Toolgen had the world's first genome editing technology to apply CRISPR-Cas9 to eukaryotic cells. Toolgen has been first immersed in the therapeutic field, developing various valuable therapies, and has recently entered seed research and business development. One of the missions of Toolgen is to secure sustainable food, and here we will introduce activities of Seed R&BD. The Seed Research Center was opened in October 2018, and since then, the business unit has joined and changed to the Seed R&BD department. Currently, the department is located in Seoul, but in May 2023, it will move to the newly built R&D Center in Osong, about 150 kilometers south of Seoul. The total area is approximately 20,000 meters square, of which 5,000 meters square is the building where we will place laboratories, offices, culture chambers, and indoor greenhouses. Several activities are underway now with the Seed R&BD. First of all, using CRISPR-Cas9 technology, we are developing new genetic resources, which we've been working on for about 4.5 years, starting in 2018. Second, we are growing genome-edited crops through a joint venture with a Chinese biotechnology company. Third, in 2022, a corporation was established in Central Asia to cultivate, produce, and test various seeds. We plan to sell ordinary soybean seeds first in 2023 to establish a market network. Fourth, we have partnerships with domestic and international companies and university research institutes for collaboration. When we apply the genome editing technology to plants, the transfection of the editing components, for example, plasmid vector or RNP, to the plant cell is the biggest obstacle because those components must go through the cell membrane and find the complementary DNA sequence in the host genome through the nucleus. Scientists at Toolgen mainly use 3 transfection techniques. The first strategy for transfection is agrobacterium-mediated transformation. A plasmid vector with guide RNA and Cas9 gene transforms the X-plant by agrobacterium infection. And we could select the genome-edited plant through induction of callus and plantlet regeneration and NGS analysis. The second strategy is the transfection of RNP to the protoplast. We isolate protoplast, incubate with RNPs, do protoplast culture, and get a regenerated plantlet. And finally, we select the genome-edited one and get seeds for the next generation. The third one is microspore transfection. Compared to obtaining the regenerated plantlet through callus induction, embryo regeneration is easier to get embryo when microspores are cultured. We are working on an embryogenesis process from the microspore transfection to get regenerated plants, although the efficiency of microspore transfection is low. The Seed Research and Business Development at Toolgen is developing high-value added varieties for productivity, environmental stress tolerance, and pest disease resistance using CRISPR breeding technology to solve the food security threat of humans and livestock. Generally, genome-edited varieties go through several stages before they are commercialized. The entire process before commercialization takes at least 6 years, depending on the crop. For Phase 1, we can obtain seeds from the selected editing crop with no foreign gene insert. Phases 2 and 3 are selection processes undergoing breeding practice, field testing, and small-scale seed production. We are conducting several research projects and have selected a soybean variety that has undergone this process, and the soybean will be commercialized first. Here is some research data we obtained through genome editing, and we want to share with you what interests us. Firstly, we have developed soybeans containing more than 80% oleic acid content in seeds, while the control group comprises about 25%. This content is higher than the oleic acid content in olive oil, and the oleic acid content is constant in soybeans regardless of the cultivation areas, local and abroad. This data shows genome editing of a specific gene causes soybean fatty acid content partitioning from linoleic acid to oleic acid. Secondly, we have developed browning inhibiting potatoes by editing the gene that causes browning when the potato skin is removed. This editing was done by RNP transfection to the protoplast. Surprisingly, the browning inhibiting character lasted up to 2 days at room temperature. Thirdly, we have generated genome-edited corns targeting herbicide tolerance, and selection experiments will be conducted this year. Fourthly, we have also obtained genome-edited chili pepper targeting drought tolerance, and selection experiments will be conducted this year. Fifthly, Cas9 overexpressing genetically modified chili pepper was successfully obtained. We will use this GM pepper for identifying the gene function study by inoculating a viral vector with a specific guide RNA to Cas9 GM pepper. The edited pepper in the next generation can show a visible phenotype caused by the guide RNA target, so this tool will play many roles in the functional analysis of pepper genes. We applied the regulatory exemption to the USDA APIS for 3 plants shown in the table. The USDA APIS approved the GMO regulatory exemption of the petunia in April 2020 and the soybean in July 2020 under the MI-regulated system. The patents have been registered locally and overseas. Also, the USDA APIS Secure Rule Scheme approved the GMO regulatory exemptions for the browning suppressed potato in September 2022. Toolgen registered this potato to a Korean patent and foreign patents that have been filed. There is a need to mitigate national biosafety management, such as current risk assessment and various approvals, for genome-edited organisms that are precisely modified to resemble traditional breeding or natural mutations compared to existing genetically modified technologies. To harmonize with international regulatory trends in genome editing, the Korean government submitted the LMO Amendment Act to the National Assembly in June 2022 as government legislation, but the bill is still pending. The new act applies the following: new exemption regulations for risk assessment, importation, production, and use approval are applied to the products obtained by genome editing technology, whose safety has been confirmed at the level of traditional breeding or natural mutation. Therefore, the LMO regulation exemption is possible for genome-edited crops through the preliminary review. However, the genome-edited organism is LMO under the Korean LMO Act of 2008. There is currently total opposition to this vague new legislation in Korea. If the genome-edited crop is LMO, this amendment is ineffective in industrialization, so industry academia research institutes oppose the current revision. Meanwhile, NGOs and GMO opposition groups in Korea oppose this bill because genome-edited crops are LMOs and should not be exempted from the regulation. Another activity that Toolgen is working on is customer service. The seed business team conducts, firstly, the synthesis of single guide RNA and supply of Cas9 protein. Secondly, single guide RNA validation of the target gene using protoplast. Thirdly, NGS service. Lastly, the construction of genome-edited crop. ToolGen's mission for seed research and business is to provide better germplasm to breeders, so breeders introduce elite varieties to farmers. ToolGen's effort would lead to higher food and feed production and sustain a better environment and ecosystem for global wellness. Please let us know if you are interested in the ToolGen Seed Research and Business Partnership for collaboration. Thank you for your attention.